This video is going to cover the various levels of gene expression and rather gene regulation in eukaryotic genes. All right, so the first thing I'm going to do is I'm going to go through one slide and I'm going to summarize all the basics that you need to understand for the test, for the final. And then you can stop there. If you want to keep going, I'm going to go into some more details in each of the various ones. And you can feel free to cruise through this and pick out what you need to understand. So the first level of gene control that we're going to talk about is chromatin structure. Okay, and so this is at the broadest levels. This is how DNA is turned on or off in terms of the broadest scales. Okay, so entire chromosomes can be dealt with like this. And what we're going to talk about here are histones, okay, and how histones can be modified, and then the DNA sequences themselves directly. All right, so here's how it basically works. A histone is a protein that has a little tail on it, and the tail's positively charged. Okay, remember that DNA is negatively charged, and so these are just spontaneously going to attract each other. All right, so if you have a histone, a group of histones that have their positive charges, they will spontaneously bind the DNA and they will wrap up the DNA. Okay, so the DNA will be tightly bound. There's your heterochromatin. Okay, your heterochromatin. This is the state of genes being off. That's the simplest way to think of it, okay? Basically, if you hide the promoter region of a gene, right? So remember, genes have promoter regions and then the transcription unit, right? Remember that RNA polymerase will bind here after transcription factors bind and activate the gene. If you hide all this by wrapping it all up, you shut the gene off, okay? So that's the simplest way to control gene expression. Okay, so this is the first level. Now, how do you decide if the DNA, which is negative, is binding to the histone, which is positive? Okay, the key is regulating that positive charge. All right, and so there are two enzymes, histone acetyltransferases and histone deacetylases. Okay, a histone acetyltransferase will, so a HAT protein, will transfer an acetyl group to that positive charge, masking it. If it masks the positive charge, if this is now essentially neutral, okay, then the histone will no longer spontaneously bind to the negatively charged DNA. It will fall off, opening up the chromatin, right? And so we could say that the promoter is now accessible to transcription factors, okay? So you can say the DNA is crumpled up, okay? The HAT protein will then open it up. And so now we can activate promoters, for example. All right, so you would consider a HAT protein as an activator, would turn their genes on, right? So HAT equals an activator. All right, the opposite of this, the deacetylase, this is where we take the histone that's been acetylated. Okay, so this is the histone protein, this is the tail that has a positive charge. That's an acetyl group, so it's being masked. And so it's not going to bind to the DNA, which is negatively charged. All right, and so this is, therefore, the gene is on because the DNA is available for transcription factors to bind. A histone deacetylase will remove that acetyl group, exposing the positive charge, allowing the histone to bind to the DNA, and thereby shutting it off, hiding it. Okay, so HDACs, or histone deacetylases, are repressors or suppressors, depending on how you want to say it. All right, they turn genes off. All right, so you have the HAT and you have the HDAC. It's like yin yang, and these guys are the two opposing forces that decide if DNA is available, right, negatively charged with the histones fallen off because they're acetylated from the histone acetylases, or if the DNA is tightly bound to the histones because the positive charge of that tail is available to interact with the DNA. All right. The second way we can regulate chromatin structure, simply put, is by methylating the DNA. Now, if you remember, a methyl group is just a carbon with some hydrogens. All right, and the key here is this is hydrophobic. Hydrophobic. Remember, hydrophobic means it doesn't like water. So if we take some DNA, which is negatively charged, again, if it's negatively charged, it likes water, right? It'll bind to water, it'll form hydrogen bonds. In other words, the DNA is soluble in water. Okay? If we methylate DNA, if we have areas here where we methylate it, these become hydrophobic pockets, and it ends up crumpling up the DNA from the water, taking it out of play. DNA methylation tends to be a more permanent solution as opposed to the histones. Okay, so DNA methylation and histone acetylation are two ways that we can open and close chromatin structure. Epigenetics is simply the inheritance not of DNA sequences like the A's and the T's and the C's and the G's getting passed on, but the patterns of which the DNA are methylated or acetylated, acetylated. The idea here is the patterns of gene expression are inherited. And so that's what epigenetics is. Okay, epigenetics is basically inheritance beyond the sequence level. Okay, so that's chromatin structure. The second level here is transcription control. This is more generally considered the primary means in which we regulate genes. When we talk about gene regulation, we're generally talking about transcription regulation. Now, whether or not that's the most universal way it's done, or if it's because it's the easiest one we've learned about and we know the most about, that remains to be seen. But in either case, transcription control tends to be synonymous with gene control or gene regulation. And you've already learned the basics of this. There are basically two kinds of proteins that activate a gene. So if this is a gene, if this is the promoter region again, and this is the transcription unit, right? So these are the exons and the introns. There are proteins that bind to the promoter. Okay, and these are called transcription factors, so we'll just call them TFs. And transcription factors, in turn, when they bind to the promoter, recruit RNA polymerase II. Right? Remember, RNA polymerase II is unable to bind DNA directly. And so this is a really good way to control it, by swarming the promoter with a whole bunch of transcription factors that then basically create a landing pad for the RNA polymerase. All right, so you can imagine a protein binds to the DNA, which in turn binds to another protein, which may bind to another protein, and so on and so forth, producing this, what we call the pre-initiation complex. And then the RNA polymerase will come in here and bind. All right, so you would consider these proteins activators because they turn the gene on. All right, in contrast, you can imagine a repressor would be a protein that's bound here, and it prevents other proteins from binding. And so the gene can't be turned on. It would repress transcription or suppress transcription. You'll see the word suppression or suppressor or repressor interchangeably, depending on where you're reading. So a suppressor protein or a repressor protein prevents DNA from having the activators bind. All right, that's one way the repressors work, by binding to DNA itself and preventing other proteins from binding. Alternatively, you can have an activator bound and then a suppressor bound to it, preventing proteins from binding to the activator. Okay, so that's a simple example of activators and suppressors or repressors. And there are hundreds and hundreds and hundreds of different transcription factors. And so remember that transcription factors bind to some region of DNA, right? Or you might remember the TATA box, the TATA box. And so there's the transcription factor TATA binding protein that binds to TATA. And then TBP in turn recruits other transcription factors. Okay? Some transcription factors are generic and general to all promoters. And so those are called housekeeping genes. And some promoters, housekeeping genes. So housekeeping genes are controlled by a broad array of universal controls, general transcription factors. And then some genes are only on in some cell types, or they're only on when you need them for certain things. And so those are specific transcription factors. And specific transcription factors bind only if their binding site is present within that promoter. Now your book distinguishes these kinds of elements here. These are called control elements. All right, so control elements. A control element is just some sequence of nucleotides, I just made that up, that will bind to a protein in a sequence-specific fashion. Okay, so for example, if this word is right here, we would consider that word a control element. All right, and because this control element here happens to be pretty much near the promoter, or really I consider this in the promoter, these are called proximal, because they're close, proximal control elements. Okay, now here's the thing, if we go all the way down really, really far over here, there might be another sequence over here, far away, a distal control element. And a distal control element is not considered part of the promoter. There's actually, if there's a series of them here, we would call this region here an enhancer. All right, so you can say that there are enhancer sequences and promoter sequences. That's the easiest way to do it. So if this is the whole, let's say that this is the gene over here. This is the transcription unit. So this is where the introns and exons are. Right next to that, we would have the promoter region. I consider promoters as containing the proximal control elements your book talks about. Okay, proximal control elements. And then really, really far away over here, there'll be some series of sequences we'll call the enhancer. And the enhancer is a collection of distal control elements. All right, now what are they doing all the way over here? Well, remember, DNA is three-dimensional, and this is existing within a three-dimensional area. So you can imagine this is a big spaghetti noodle, right? And so watch this. Whoa, there we go. Now we have the enhancer hanging out right next to the promoter. All right, and so that's how they can impact the transcription of specific genes. It's also important to note that enhancers are generally specific to one gene. All right, so if a gene has an enhancer, that enhancer is for that gene, not another gene, not somewhere else, but for that gene. That's the current understanding. Okay, a transposon, or a transposable element, this is crazy. Ready? There's some DNA. Let's say that right here is a promoter region. And somewhere way far, far away, maybe even on a different chromosome, there is some sequence of DNA that will get cut out by enzymes. It will literally get snipped out, and so that DNA is hanging out now. It will then go and it will randomly insert itself into DNA. Yes, so there are elements of DNA that will cut and paste themselves out and back in again to the genome. If that happens to occur inside a promoter region, so if this is the intact promoter, okay, and so the gene's over here, if the transposon inserts itself into the promoter region, you've now broken it, right? So this is, let's call this the transposon, just transposon, TO. Transposable element, TOE, or just TE. It doesn't matter. This is a sequence of DNA that came from somewhere else, and it interrupted the promoter region, okay? And so as a result, this gene is now off. And then some other time down the road, some generations later, this transposable element will cut itself back out again, restoring function to the promoter, and so it's back intact again. Okay, so that's a transposon. And so those are the two examples of what we would call transcription control. Right, remember that at the end of transcription, you're left with a primary transcript that contains both the introns and the exons, right? And so of course the next thing we're going to talk about is post-transcriptional control. And we've already covered alternative splicing a bit and variable poly A tails. We've talked about these in class enough, so I'm not going to spend too much time here. But the key, one little insight here, is understand that everything past this point technically is post-transcriptional. All right, but in terms of understanding the different levels of eukaryotic gene regulation, you have to understand that post-transcriptional control is limited to that processing step. within the nucleus, right? So you can picture everything up to this point here has happened in the nucleus. We've had the chromatin unpackage, we've had transcription occur, we have a primary transcript. Okay, so that's transcription control. Now we're going to cap and tail that primary transcript. We're going to splice out those introns. We're going to have what we call a mature transcript, right? And then the last thing we're going to do is export it from the nucleus. So there's three ways really. The last thing here I want to add here is to modify nuclear transport. I'll explain that in a second. Or nuclear export, I should say. Nuclear export. That's going to be another way to control this. So alternative splicing we talked about, right? That's the example where if we have exon 1 and we have intron 1, we have exon 2, we have intron 2, we have exon 3, right? So we have these various regions here, and then we have our poly A tail. All right, you can imagine, so one splicing would take out, there's the first intron, there's the second intron, right? And so there's exon 1, 2, and 3. You can see how that would lead to a primary, or a mature transcript that has exons 1, 2, and 3 on it with the poly A tail, right? And then the cap. However, if we leave exon 2 in with a loop, so in other words, if it looks like this, here's intron 1, okay, here's exon 2, and then here's intron 2, and so this is exon 3 and exon 1. You can see how that's going to now give us a shorter transcript where we just have exon 1 and 3 with a poly A tail. Okay, so this is alternative splicing. And this is another means of regulating gene expression. Okay, variable poly A tails, the name says it all. You can see here's the cap, here's the codon, here's the spliced final product, and then there's some poly A tail. And that poly A tail could be really long, or it could be really short. The really long tail, remember that there are these enzymes called exonucleases that are like Pac-Man, and they're just going to eat away at all of this. So it's like a fuse. The longer the fuse, the longer the RNA lasts. It's pretty much that straightforward. Okay, and then the last one I talked about was nuclear export. And so you can just imagine the nuclear export. You can imagine once you have this mature transcript, you got to get it out of the nucleus. Well, let's say that you don't. Let's say that you shut the door. Okay, you shut the door right here. So now you're regulating it at that level. That's still considered post-transcriptional control. Okay, so now we get to the next one. This is translation control. Okay, so at this point, we are in the cytoplasm now. All right, we are now post-transcriptional, and now we're translational. So this is all about, this is all about the ribosome binding. Okay, now much less is known about the level of translation control, so we're not going to talk much about it other than this. Here's the 5' cap, here's the poly A tail. Okay, and we're going to talk, here's the start codon. Let's see right there, there's your AUG. Right, so that's where, if you remember, the small subunit is going to bind, and then the initiating tRNA will bind, and then the large subunit will bind. And the large subunit will bind, right? Sorry, I just had to clear my throat. Anyway, remember, just upstream of this, on the 5' end, there's the untranslated region, or UTR. Right, and remember there's a 3' UTR as well. So again, UTR means untranslated region. It's a region that does not have codons, but it's still part of the mature transcript. And so you can imagine, if this is the transcript, and this is the UTR, let's say right here, that a protein can come along and bind to the 5' UTR. And that protein happens to block the small ribosomal subunit from binding. And so you've just shut down translation. Okay, so we'll just leave it at that. There are translation inhibitor proteins that bind to the 5' UTR. All right, there are others. There's 3' UTR situations as well, but we're not going to talk about those. All right, so that's translation control. That's not post-transcription control. Okay, so remember, even though we're after transcription here, post-transcriptional control only takes into account the stuff happening within the cytoplasm. I'm sorry, within the nucleus, right? That has to do with the capping and the tailing and the splicing and the export. Translation control is all about making that polypeptide. Okay? And now we get to the last stage here, post-translational control. Okay? Post-translation control occurs after we have an active protein. The protein's been folded up, it's in 3D, it's ready to go, and now we can do all sorts of things to the protein itself. So one thing we can do is modify the protein. We can add things to it. You've already learned one of these. Throughout the semester, we've talked about phosphorylation, protein phosphorylation. You might remember CDK proteins. You might remember a kinase. A kinase is what again? Think about it. Right? It's an enzyme that adds phosphate groups to its substrate, whatever it is. All right, so that's an example of a post-translational modification. One simple example to think about is a promoter region where there's a protein bound to the promoter, and on that protein is another protein that's hiding the transcription factor. So let's say that's an activator, and then the thing binding to it's a repressor. Okay, so in this example here, this activator protein is called an E2F. And the repressor protein here is called RB. Okay, the protein product of the retinoblastoma gene. And RB sits on E2F and it just shuts it off. It prevents it from binding to anything. However, CDK comes along, CDK2 comes along, and that kinase phosphorylates the retinoblastoma protein. When that phosphorylation occurs, it removes the retinoblastoma protein, allowing E2F to just sit there and recruit what it needs to. And actually E2F also gets phosphorylated, which helps it do its job. All right, so phosphorylation is a very important example of post-translational modification. All right, if you remember all the way back at the beginning of this, we talked about HATs and HDACs. Okay, so acetylation is another example of a post-translational modification, where we're taking that histone protein, right? Remember, the histone itself, the proteins that model chromatin, at the end of the day, there's a gene for the histone, there's the RNA that makes it, there's an RNA that controls the directions on how to make a histone, all right? And then the ribosome makes histones, and then the histones go back into the nucleus and do their thing. And so those histone acetylations are considered post-transcriptional modifications, all right? And then lastly, there's a protein called ubiquitin. And ubiquitination is adding ubiquitin to a protein. So ubiquitin itself, all right, so hey, if this is a normal protein, just a generic protein hanging out in the cell, ubiquitin is a small protein that gets added to it. And you know what? It's kind of like a flag saying, hey, I'm here. And what the ubiquitin does is it recruits this big machine called a proteasome. And the proteasome is basically like a recycling bin. Proteins come out one end and amino acids come out the other. And these things are hanging out all over the cell. They're called proteasomes. I'm not sure why general biology skips these as part of the cell biology aspects, where you talk about things like the lysosome and the ribosome, but for some reason you don't talk about proteasomes. But you will definitely run into this again if you ever take cell biology, molecular biology, or any of the higher level biology courses that relate to the inside of a cell. All right, so you have proteasome degradation. Proteasome degradation is mediated by the protein ubiquitin. Okay, that's what ubiquitin's main job is. And the reason we call it ubiquitin, if you've ever heard the word ubiquitous, ubiquitous means everywhere. So when we first discovered this protein, we found it was everywhere, and so we called it ubiquitin. Okay, all right, so that's the gist of eukaryotic gene regulation at the various levels. You can stop here if that made sense already. Otherwise, I'm going to go through the rest of these slides and just make sure that I hit on all the important points. Okay, so chromatin structure. Basically what I'm going to do here is I'm going to take each one of these big headlines and dig into them. All right, so this slide talks about histone acetylation. This talks about how it opens up chromatin. This talks about that euchromatin versus heterochromatin relates to the state in which the chromosomes, the chromatin is present or not with acetylation. Right, so again, if we have open chromatin And so this is like the on state that would correspond to euchromatin, the good chromatin. And the reason it's open and on is because the histones are off of the DNA. Okay, so this might seem counterintuitive to have the histones off the DNA so the DNA can be on, but that's what's going on here. All right, and so here you can see in this picture here, this is from your book, right? So these purple things, there's actually 8 of the histone proteins, and you can see there's 1, 2, 3, 4, and then you can see hiding behind each one of them there's another one for a total of 8. You can see them better here. And notice that each one of these proteins has a tail coming out. So here are your histone tails. These histone tails are positively charged. Okay, and so that's what's causing the reaction with the DNA. That's why it can stick to the DNA, which is in blue here. All right, so when we acetylate these tails, when we bind an acetyl group to these tails here, you can see these in green, this opens up the DNA. So now you can see that it's looser. Okay, and so that's the basics of acetylation. And then methylation, as I said earlier, this is adding methyl groups to specific base pairs, or really just specific bases. And usually it's guanine. Either guanine or cytosine, I can't remember. But they're basically, within the double-stranded DNA, they're GCs or CGs, doesn't matter. And we refer to these things as GC islands. And so these regions of the genome that are rich in GC, they tend to be the ones that get heavily methylated. Okay. All right, and then for epigenetics, we talked about earlier. So the key concept here is that chromatin modification doesn't change DNA sequences. All right, these modifications in turn can be passed on. So the formal definition of epigenetic inheritance is inheritance of traits that are transmitted by mechanisms not directly involving the sequence of DNA, the nucleotide sequence. And so, for example, a gene is repressed by methylation, and then this methylation pattern is passed down to the next generation. Okay, so that's epigenetics. All right, now when we talked about transcription initiation, right, so when we talk about transcription control, we talked about those control elements. And so here's a picture from your book. Now again, your book says this about promoters versus control elements. The promoter region here, you can think of that as the area that specifically binds to fundamental basic transcription factors that then recruit the RNA polymerase II enzyme. Okay, and then very close to the promoter, we have these proximal control elements that bind to other transcription factors, specific transcription factors. Okay, other books, other people, myself included, would refer to the promoter as this region here that contains the basics and also any specific control elements. Right? And in contrast to that, really far this way, so that's why you see this double bracket, that means really far away, there are these upstream control elements we call distal control elements. And if we have a group of them, we call this whole cluster of them an enhancer region. Okay? So this is the basic anatomy of a eukaryotic gene right here. And the key is in addition to the exons that contain the 3-letter word codons, in addition to a promoter, in addition to the, basically, in addition to codons, there are other words in DNA that don't just code for amino acids. They are binding sites for specific proteins. And we call those collectively, you can think of those as control elements. More generically, we talk about these sequences of DNA as called a consensus site. Really, all a consensus site is a sequence of specific nucleotides that in turn bind to a protein of some kind or another. Okay? And so the key here is that these control elements, these distal and proximal control elements, they are precise, they are critical for the precise regulation of gene expression in different cell types, right? If we just had one cell type, we would have much less need to control everything. But because we have 200 different cell types, 20,000 genes, we need to have a very complicated control system here. And so you can start to see how that would build up. Okay, so I said all this earlier, I'll say it again. RNA polymerase can't directly bind to DNA, and so it needs help to get there. It requires these things we call transcription factors that initiate the process of transcription. And there's basically two general types of transcription factors. There are general transcription factors and specific transcription factors. All right, so let's go through those really quick. A general transcription factor is a transcription factor that is essential for all protein coding genes. Okay, and so some of these factors bind to that TA box, right? And many of these general transcription factors bind to other proteins that in turn are bound to DNA. All right, so again, not all transcription factors have to necessarily bind to DNA directly. So let's say that there are a couple DNA binding proteins here that then have regions of the protein that then recruit other proteins. These proteins here could be considered transcription factors too. All right, and so I like to think of it as there's a swarm of proteins that bind to DNA that all bind here that ultimately then recruit that RNA polymerase 2. Okay? All right, and an example of a general transcription factor we've talked about before now is the TATA binding protein, or TBP. And about 50% of all genes contain that TA box or something similar to it. The other 50% tend to be genes that are not universal, but instead are specific to specific cell types, tissue types, or cell types, or situations. And so now that would be about talking about specific transcription factors. Okay, so specific transcription factors, these are for genes that are not always expressed, right? So they're either cell specific or they're induced and repressed under certain stimuli. And specific transcription factors, they bind to various specific control elements. Okay, again, these are that non-coding DNA sequences. Some of these control elements are nearby, and so we have proximal control elements. Those are the ones that you could think of as part of the promoter itself. And some of them are far away, the distal control elements that together form groupings that make up things called enhancers. Okay, and then again, proximal control elements can be generally referred to as part of the promoter region. And one simple example of a specific transcription factor, one that I used to work on in grad school, was E2F, right? And recall that we had those proteins, the cyclin-dependent kinases. And one of the things these cyclin-dependent kinases do is phosphorylate proteins like E2F, okay? Activating the genes they're involved in. So this specific specific transcription factor E2F generally activates genes, so it's an activator, and it activates genes involved in S phase of the cell cycle. All right? And so activators, these are any protein that bind to either enhancer or promoter regions or distal or proximal. It doesn't matter. Your book, again, just refers to an activator, something that binds to distal control elements. But really, an activator is just anything that turns on transcription directly. And so an activator protein can bind to the promoter region or can bind to the enhancer regions upstream. It doesn't matter. The key is they have two kinds of domains. And remember what a domain is. A domain is a region of a protein. Okay, and so there's one domain that will bind to DNA, and then there'll be some other domain that binds to, say, another protein. Okay, so that's an activator. And so here's an example one. Here's a real-world example one. This is MyoD. This is a transcription factor involved in the differentiation of muscle cells, of skeletal muscle cells. And you can see here, this is the folded-up polypeptide. Right? You can see it's actually a dimer. Dimer meaning there's two of them. So you can see this orange one here, and you can see this purple one here. Okay, and so from a cartoony version, it'll go like this, and then loop around, and then go like this. Right, so this is one polypeptide, and then there's a mirror image one on the other side of it. And so this is so this overall shape here, you would think of that as the quaternary structure of the protein. This shape here, just a single part of that would be your tertiary structure of the protein. And then here's an alpha helix, there's another alpha helix, and those are all your secondary structures. And then if I went in and I looked at the individual amino acids in here, that would be the primary amino acid structure, right? And so taken together, this overall quaternary structure here has two domains. The first domain, you can highlight down here, we would say is the DNA binding domain, because these helices are at perfect angles to fit right within this minor groove of the DNA helix. And then up here, this area here, called the activation domain, this would be an example of some other protein that bound, say here, which then in turn would recruit RNA polymerase, or maybe it's even RNA polymerase itself, right? So that's the basic idea. I'm sorry, I keep having to, the top things have the animations left over from when I pasted them in before. I should shut that off. But here we are. All right, so let's see, repressors. Okay, so a repressor protein or a suppressor protein is, so let's say that carefully, a transcription suppressor protein or a transcription repressor protein. These are proteins that inhibit the expression of a specific gene, okay? And that can occur in several different ways. So for example, some might act indirectly by influencing chromatin structure, right? And so you can think of things like Histone Deacetylases. Right, so that would be a negative transcription regulator. Some of them might bind to actual DNA sequences, right, preventing the activator proteins from getting in there. And that's very reminiscent of the way we thought of enzyme control, the competitive enzyme inhibition. And then lastly, others might bind to activators that are already bound to DNA. So again, if this is an activator protein, we can think about the repressor binding here and preventing things from binding to it, right? And so again, the example there is the retinoblastoma protein, right? So that's transcription control. All right, now here's the take home to all of that. By adding up all of the different control elements, by having several different control elements, we can now start to turn genes on and off in a very complicated way, right? And so, for example, with about 12 different control elements on a single, within the chromosomes, within the entire genome, that results into a huge number of potential combinations possible. Right now, I can't tell you how many different control elements there are in the human genome. I can tell you there's probably way more than 12. All right. Okay. And this gives you the idea here. Let's say here's a gene, and here are some control elements on that gene. And somewhere else in the genome, there's the same 3 on this gene. And so this gene and this gene, even though they're nowhere near each other, because they still have the same control elements, will bind to the same transcription factors and will be activated at the same time, reminiscent of the eukaryotic, of the prokaryotic operon. And so I like to call these things gene constellations, because if this is a nucleus, you can imagine a bunch of dots lighting up, and all of those dots would be all of the different transcription factors binding to the promoters and all those genes all at the same time. Okay. And so here's a simple way to think of it for those of you who are more visual. Here's 3 control elements. They're just arbitrary sequences of DNA, and we're just making it up here. Okay. And so each control element can either be off or on, right? And so that could be represented, if this is control element 1, for example, either the protein's bound to it and it's active, or the protein is not bound to it and it's not active. All right. And so with just 3 control elements, right, each one being either on or off, okay, I should say either on or off, we can have a total of 2 different combinations for each, which gives us a total of 8 different possible combinations. And so this gives us 8 different ways of controlling genes. Okay, so for example, you can have it where all 3 are off, all 3 are on, or one or more of the others is on and off. Okay, and so this is just for 3 elements. If we had 12, okay, that would be 12, that would be 2 to the 12th power. Okay, so that's a massive amount of combinations. And so what this does is it gives a complex organism like humans, where we have over 200 different cell types, we have about 20,000 genes, we can control all of these genes in very complicated patterns, resulting in the complexity that we see in all multicellular life. Okay? All right, you can watch this on your own. This is just an animation showing the And let's see here. We can, okay, so hopefully you heard that. If not, feel free to just download the PowerPoint, listen to it on your own. Okay, so transcription alone does not constitute gene expression. Regulatory mechanisms can operate at various stages after transcription, and such mechanisms allow a cell to fine-tune gene expression rapidly in response to an environmental change. Okay, and so that just summarizes that whole idea of differential gene control using multiple elements. All right, we already talked about alternative splicing. This slide just summarizes that. So the one little take home here is that there's about 90% of all human protein coding genes at one point or another undergo alternative splicing. Okay. You can watch this on your own. I'm not going to play this right now. This has to do with mRNA degradation in the poly A tail. This is alternative splicing of the troponin T gene. And one little extra here, and you should know this, is that isoforms, so protein isoforms are families of proteins all derived from a single gene. All right, so you can see this RNA here would give rise to protein A, and this RNA here would give rise to protein B. And so you would say protein A and protein B, that's an isoform family. Okay, you would say protein A is an isoform of the troponin T gene. That's how you would talk. Okay. All right, here's another video you can watch. This has to do with the regulation of translation control, and this has to do with those mystery proteins binding to the 5' untranslated regions we talked about. Okay, and then this talks about ubiquitination. This talks about your proteasomes. All right, so you can skip through that. Protein processing, your book does a little bit more detail here than I did, but basically some proteins, so here's a folded up polypeptide chain. You see these little lines here, those are your disulfide bridges. So this would be an amino acid that has a sulfur group as its R group, and so with this one, and so basically so this is an amino acid that has sulfur, and this is an amino acid that has sulfur. This is called a disulfide bond or a salt bridge. That's what you're seeing here. These stabilize structures. All right, and so what could happen here is we can actually cleave the protein here. All right, and we can phosphorylate it over here, and you can see here. This is, just watch the video. All right, so there's many things you can do after the protein is translated, basically. And so collectively, we can talk about these as post-translational modifications. Okay. And then this animation talks about ubiquitination. You don't have to really know too much about that, other than proteasomes degrade proteins back to their amino acids. Okay, and so that's eukaryotic gene regulation in a nutshell. And I will stop this video and get this uploaded as soon as I can. The rest of this PowerPoint we already went over in class, so you don't have to, I'm not going to sit here and read through all this, but this is basically all the series of point mutations that you guys are expected to know. Okay? All right. I'll see you guys soon.